Welcome you all for another session of RNA biology. Today we will resume where we left in the previous class, and this was the last slide which we were discussing, that is about the Murchison meteorite which was discovered in the Australia, Queensland, Australia. So Victoria, Australia in fact, and different compounds are measured using various chemical techniques, and there is a relevance why this was compared to Murchison meteorite. So before going into the details, I would like to introduce the teaching assistants who would be assisting me in carrying out this lecture series. Is Mr. Ashish Sethi, Ms. Sharanya Premraj, and also Mrs. Ansi Chowdhury. So these are final year PhD students who would be part of conducting this lecture. So now in the previous class we had discussed about Urey-Miller experiment, one of the major finding of RNA biology research. So in 1953 at University of Chicago, they conducted a very simple experiment, as you can see here, in a big glass container where you have water vapor, and also you have given two electrodes, and also different gases such as methane, ammonia, and water vapor and hydrogen. They are there, and the electrodes are kept such a way that they can be given a powerful electric shock. And what happens is, because the water vapor etc. are present after this discharge experiment, then you have a condenser below where you have a cooling option, and the contents, what this electric discharge, and also when these inorganic gases and water vapor, hydrogen etc. There is a reason why these molecules are selected, because the prebiotic world is mainly supposed to contain these molecules abundantly, unlike modern times oxygen etc. So after this solid discharge going on, what you end up getting is the products that are formed because of this very high temperature and also very high electric pulses, and then they are conducted in the bottom. And needless to say that there is a source of water vapor which is there below, which is heated, and which is a water molecule which is boiled, and the vapor will be discharged into this chamber. So now they analyzed the component with that of the Murchison meteorite. So that is something very interesting. If you look into the amino acid composition like glycine, alanine, etc., you can see they are equivalent in amount. That is, 4 dots means abundant, 3 dot means reasonably abundant, 2 dot means present, and 1 dot means very scarcely present. So this is the density or the intensity representation of individual components, and I will not go into the details of each and every component. So whenever you compare the components that were present in this Urey-Miller experiment and to that of the Murchison meteorite, they find there is a reasonable similarity between the components. So what it says, that the meteorite, when it is entering the atmosphere, that is of course coming from a outer space, and this very high temperature and interaction with the various gases etc. can naturally allow the formation of these components. So what we should understand, the nature did not choose a specific compound or a specific chemical for the life to originate. Rather, it used the chemicals available then and the product that are formed because of the prebiotic conditions, and then a life is originated. So that is what this similarity indicates. So now if you look back into the building blocks of life, that is various biomolecules, and especially from the Urey-Miller or Miller-Urey experiment, and we can see a lot of compounds, that such as how glycine is formed, how formaldehyde is formed from ammonia, because various components can combine together in presence of water. It can give rise to several biomolecules. Biomolecule is little bit premature to say right now because life hasn't originated then, but eventually these molecules end up being part of various life system. So several years after the discovery, people find out that what are the conditions and situations through which these molecules got a upper hand for being part of living organism. And we will see them one by one. So one more point which we discussed was the hydrothermal vents. So what are hydrothermal vents? We know under the planet surface there is very high temperature is there, and where the planet's crust is thinner, there is a chance that this high temperature beneath the planet Earth's surface will be exposed to the atmosphere. You all know about volcanic eruption, explosion, etc. So in this picture, you can see two situations. One is a volcanic depiction and another is a hydrothermal vent. Basically, volcano and the hydrothermal vents are one and the same, except that volcano is on the terrestrial surface, that is the dry land, whereas at the sea floor, the same volcano when it comes, that is acting as a hydrothermal vent. In both cases, in prebiotic world, there are lots of inorganic substance, and this heat, intensive heat, both you see in the volcanic surface and also in the hydrothermal vent, will provide a catalyst or a accelerating agent for a biological reaction to happen. And we know even now when a volcanic eruption takes place, a lot of gases, inorganic substance come out, and because of the high temperature, it has the potential, it has the power to give rise to various biomolecules eventually, and it keeps happening. Even now you can produce various biomolecules in laboratory conditions. So not the point that volcanic environment and also the hydrothermal vents could also trigger, similar to what you saw in the case of Murchison meteorite. So now, do we have other evidence? That is what we are looking for. Okay, we are saying that these molecules could have been found near the hydrothermal vents, volcanic environment, etc. But do we have any evidence? One such evidence what we can get from ribosomal RNA phylogeny. So prokaryotes are the primitive life forms. Broadly, the living organisms are classified into two: prokaryotes and eukaryotes. So we will not go into the details right now, but understand the prokaryotes are more primitive in terms of their organismal complexity compared to eukaryotes. Hence, they are considered to be evolved much earlier on this planet. So now, when you compare different types of bacteria can be broadly classified into three groups. Archaea, which is much primitive than bacteria, then the bacteria which is actual bacteria, and then you have the advanced category that we call them as eukarya. So our interest is mainly the branch between the archaea and the bacteria. So in archaea group, you have one group called thermococcus, which is circled in red box, and then you have bacterial group, that is thermus and thermomicrobium and thermotoga. So these three groups, when you try to compare them at RNA phylogeny level, ribosomal RNA is basically used for comparing different organisms and their relatedness. Even it is used as modern days, it is used as a very powerful tool for comparing the interrelationships. So if you look at ribosomal RNA phylogeny reveal that the high temperature organism, that the thermococcus and thermus, these animals, these bacteria, they live or they prefer a very high temperature environment. And in this situation, it seems that they are more related. They have more genetic similarity compared to rest of the bacteria. So what it says that the organisms, that high temperature organism, the organism which prefers high temperature even today, they have more relatedness compared to the rest of the bacterial group, that is the mesophilic or psychrophilic bacterial group. Mesophilic means normal temperature, psychrophilic means cold loving bacteria. So thermophilic bacteria are more related and they are more primitive also. So this suggests that these organisms could have originated much early in the life, origin of life on the planet Earth. So now let us think, can we make various subunits or components of RNA. We were all discussing about the RNA World hypothesis. So RNA have got three major components. One is a phosphate, that is acting as a backbone of the RNA, and then a ribose, a pentose sugar, and then a nitrogenous base. So phosphate can come from rock weathering because there are many phosphate containing rocks are there, and they can be during rain and other environmental conditions, they can provide adequate quantity of phosphate. And the ribose sugar, you can have carbon dioxide, energy together. 5 COH2 molecule, that is basically formaldehyde molecules combined with water, can give rise to ribose sugar. We'll come to that more in detail, name of that reaction, etc. And the nitrogenous base, methane and ammonia, nitrogen, methane and nitrogen, along with the heat, can give rise to hydrogen cyanide. It's also called as hydrocyanic acid. And 5 hydrogen cyanide molecule can give rise to adenine. So what we should understand, that the phosphate can come from the rocks, ribose can come from a chemical reaction, and adenine also can come from the chemical reaction. Remember, adenine is one of the nitrogen base. There is RNA has got adenine, uracil, cytosine and guanosine. So there are 4 bases are there, and in DNA the uracil is replaced by thymine, which we will see later on what is the difference, etc. So you can see the ribose sugar usually combine with the nitrogenous base, and which in turn make use of a phosphate backbone to give rise to one monomer of the RNA. And the other bases, 3 RNA bases, are guanine, uracil and cytosine, whose molecular structure is being given here, and they also can be formed spontaneously. Means what we should understand, without any enzymes or any other new intervention, purely from the inorganic material, you can get this organic molecules such as ribose sugar and nitrogenous bases, which can eventually participate in the formation of the RNA. Now this is a typical structure of the cell, and we know that cell is circled by a plasma membrane and also a nucleus where the genetic material is situated. So this is a typical building block. But if you look further, we can understand that they are formed from various monomers, and we call them as macromolecules. Like nucleotides are monomers for the nucleic acid, and every molecule of the DNA or the RNA, they can assemble together. Usually the DNA can assemble together and it can give rise to chromosome. And same way, there are many carbohydrates. Carbohydrate monomers can group together and can form polysaccharide. And polysaccharides can contribute to the structure, and also it can contribute to the energy yielding reactions also. And then comes the fatty acids. Fatty acids are also spontaneously formed. We will not go into the details of that because this topic is about the RNA biology, and they also can assemble and they can act like a protective cover, that is the plasma membrane. And so we have amino acids, we have carbohydrates, and we have nucleic acid, and we also have the lipids formed spontaneously, which can now assemble and to act like a miniature version of the cell. Now we have to bring in the evolution theory, because evolution we kind of discussed in the previous class. That means any change can technically be called as an evolution, provided that change is adapted, that is welcome into the community or into a society or into a environment. Then that will have a tendency to be selected, that will have an affinity to be preferred. Like I gave you example of a mobile phone, evolving mobile phone, different features, etc. If it is adapted for the environment, then it will be selected, and then we can call it evolved. So we can think various similar examples. So we have to bring in evolution theories every now and then. So let us bring evolution theory. So the unity of life, all existing living forms can be formed only from the pre-existing form. This is a non-fact. However, it makes use of the materials available in the environment. Say for example, if you have a bacteria, and bacteria simply cannot make a copy of itself from nothing. it has all the information, all the resources for making another bacteria. However, the resources has to come from the environment. So a bacteria is formed from the same material, and it functioned according to the pre-existing bacteria or pre-existing cell. And all organisms are descendants from a single ancestral form of life. This Darwin also says, and various evolution theories also says, there is no way every organisms are formed independently. Of course, they are evolved independently, but the so-called life form must have originated in one form. And what are the need of life? The life always needs a genetic information, then energy and catalysis. So these are the three fundamental steps for the propagation of life. And continuing the evolution theories in a different angle, we should ask some questions. What is the manner in which the ancestor emerged from materials available then? Can we reconstruct it? This is a $1 million question. Why? Because whatever you put effort, it is very difficult to recreate a prebiotic world unless you go to a planet where there is no life exists and start doing experiment. Because no matter what you create in the lab condition, they are always the miniature version. The problem with the miniature version is the area, the volume and the resources are limited. When you talk about a planet, you are talking about large scale, and the scale sometimes is very important, because scale and time which we cannot create in a laboratory condition. You can do proof of principle. That is why the origin of life on planet Earth is not creatable or definable in a laboratory condition. And then comes the next question: how did all existing living organisms evolve from the common ancestor? And we can justify how it originated from the existing common ancestor, but how all these forms are evolved, it requires billions of years of evolution and lot of luxury of space and resources. And coming back to the RNA world, we know RNA can store genetic information. From the modern world's knowledge, I am saying this. And RNA can be temporary copy of the genetic information. We know various viruses are there, like influenza virus, even this COVID virus. These are all having RNA as their genetic material. And RNA can act as a carrier molecule. And also, RNA can be a working part of the cellular machinery. So these are all existing even in modern day. So there is no reason to believe that there is no RNA world existed. There is always supporting evidence to suggest that RNA world indeed is a reality. And most importantly, RNA can act as a catalyst. Catalyst means capable of carrying out a biological function. Now, let us look into the RNA world using a time scale. So, around 4.5 billion years ago, you have life, or the planet Earth that existed, which is devoid of any life. And around 4.2 billion years, you have the stable hydrosphere. And by around 4.2 to 4 billion years ago, you have got prebiotic chemistry. And then around 4 billion years ago, close to that, you have the pre-RNA world. That means the RNA world is not formed, but the components of the RNA have existed. And around 3.8 billion years ago, you have RNA world came into existence. And again, after several millions of years, by around 3.6 billion years, you have the first DNA and protein molecules that are formed. And but around 3.6 billion years to modern world is the so-called life existed. So around 1 billion years, from 4.5 billion years to 3.6 billion years, around 1 billion years, this evolution was happening on planet Earth. And needless to say, when planet Earth was formed, it was too hot for any so-called life forms can exist. So the cooling process also is very important, which took place in over millions of years. So let us ask, in the modern world or modern life form, you see majority of the polymers are proteins. But can we look little bit more in detail? So far, every polymer that we know is mainly the major component is a protein. But however, Proteins are the result of polymerization of individual monomers called amino acids, and we already showed almost all amino acids can be formed from the prebiotic world through inorganic materials. Now we are learning, although we know in the modern world the predominant polymer is protein. I'm not saying that RNA and DNA are not polymers, they are polymers, but the predominant polymer is the protein. But now we are learning more in detail about a polymer that existed in the RNA world. And nucleic acids are the major polymers then, and they are formed by the polymerization of monomers, and we call them as nucleotides. And nucleotide is basically a three-member component. One is this phosphate group, and then you have a pentose sugar, and then you have got a nitrogenous base. And this pentose sugar has got 5 carbon atoms, and we usually name them as 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So you can see here, this is 1, 2, 3, 4, and 5 carbon. And these 2 and 3 are very, very important, and also the 5 prime end. And we will see them when you study more in detail about the RNA. So in the RNA world, we now know that the RNA should be able to self-replicate, and RNA replication is more plausible than the DNA replication. We will come to that, how RNA evolved into DNA and what was the necessity, etc. DNA, for some reason, because of its structural stability, etc., it always requires support from other molecules. So we have to understand, if there was a self-sufficient molecule, that should have been RNA. And there are other components such as ribosomes, which acts as RNA catalyst in every organism, starting from bacteria to human even today. And there are also a large number of RNA molecules that can be found in the modern world, even in existing life forms. Now, the most important point which we are trying to discuss in the RNA world is that how can we assume that every molecule that are formed must contribute to the RNA, or how can we assume in the prebiotic world RNA molecule should have been present or it should have been preferred? The answer is no, it is not like that, because when you have these various complex organic molecules that are formed by random chemistry, they can evolve in any direction. Direction number 1, direction number 2, direction number 3, direction number 4. But as the time passed, only one line is preferred. Say for example, let us assume one family had 4 kids. Kid number 1, kid number 2, kid number 3, kid number 4. Kid number 1 did not marry. Then there is less likely that will propagate further. And kid number 2 married, had no kids. Then second line also stopped. Third kid had married and it had kids. That organism had kids. That person had again kids, and that line is continuing. And there is a reason behind it, because that particular molecule or organism is preferred. That organism have the fitness, that have the adaptation or the ability to gel with the environment. Because of all this reason, one lineage is preferred, and that lineage is what we are having, RNA. RNA later was able to make a copy of itself, and RNA was able to later assist in the assembly of amino acid to give rise to proteins, and this RNA and the protein collaborated together to give rise to DNA, and so that's how that lineage existed. That is why there are different theories where people like NASA and other organizations will be searching for life on Earth. There are various organizations search for life on extraterrestrial planets, etc. But what we should understand, the life formed on planet is by a chance factor also. It is not that inorganic material, if there is a methane, ammonia, hydrogen and water vapor is there, life can form. Of course can form, but it need not always end up in form. So we should understand, it is not just a question of having the raw material for life. You can always end up having a life form. So it is also a chance factor. So from this RNA world, you end up having an RNP world. RNA world had only RNA. Then you have got RNA plus protein. We call it as RNP, ribonucleic acid plus protein. That is RNP, and that eventually give rise to DNA, and then the LUCA. What is LUCA? Last unique common ancestor. And this is the beginning of life in the subsequent generations. Maybe I will stop here and will address you in the subsequent class. Thank you.